All right. Hello, everyone. And very nice to see all of you here in this planetary biology seminar talk. And in particular, it's very nice to see Daniel. So let me say a few words about Daniel. He will introduce his career himself. And first of all, Daniel, it's really, really nice to see you here. Here, say in the seminar here at EMBL, here in Europe, because for others, I know Daniel from my time at UCSD, where Daniel was a postdoc, I believe, for a number of years. And I know Daniel as a person who is very passionate about mass spec, but at the same time, very passionate about basically everything. So it was really cool to witness him going through many different developments, many different applications. And I believe almost all of them, they were related in some way to either different source of planetary biology. So, and different sorts of actually extracting some mass spec or molecular information from some environmental or biological, weird biological samples. So, and this is, I know from my experience, this is very, very challenging, not just to collect data, but really make molecular sense out of this. So that's why when I learned that Daniel actually recently moved from Peter Dorrestein lab at UCSD, which is probably one of the leading labs in metabolomics for environment. So when I learned that he's moving to Tübingen, I was extremely excited just to have him closer here as a colleague, but also as a great person and great scientist I know. And so that's why we arranged this seminar. So he is a young PI, definitely hungry for projects. And I think there cannot be a better time to involve him into the planetary activities that EMBL is now kicking off and pursuing to coordinate. So with this, Daniel, the floor is yours. And I'm very, very excited also to learn about your latest updates.
Thanks a lot, Theo. Yeah, thanks so much for the very kind words and introduction, and also for actually putting me in contact with Pau and Josipa. And yeah, thanks for organizing the seminar and having me here. So yeah, before we go on like a planetary scale today, maybe just a couple of words. So my background is in actually biochemistry and natural product chemistry / analytical chemistry. And before I dove into like environmental systems, I was working like mainly like in vitro with like one particular molecule. And yeah, I just want to like show it to you quickly because I think it's a really cool molecule, very terrestrial. It's albicidin, a natural product made by a sugarcane pathogen. And yeah, I spent a lot of time with it, elucidating the structure together with my coworkers, as well as like its biosynthesis. What makes it also probably pretty interesting for like a broader community is that it has very good antimicrobial activities, both against gram-positive and gram-negative bacteria. So it's a gyrase inhibitor in the nanomolar range. And why I thought this is particularly interesting to showcase you here is because it like shows a little bit my trajectory from working with this one molecule, one gene cluster. We expressed like a lot of like the genes and reconstituted its biosynthesis in vitro to come up with like this very comprehensive model. However, at the end of my PhD, I was wondering a little bit about like its ecological role, and as it's produced by a sugarcane pathogen, I was fortunate to spend 2 weeks at my collaborators' lab in southern France in Montpellier, and we tried to extract simply albicidin directly of its natural environment here, the sugarcane plants. And what really struck my mind was the amount at which this is produced like in vivo. So we spent like years and hundreds of liters of Xanthomonas culture in order to produce enough material to like finally solve the structure by NMR. And then we realized that actually in its natural host, it's like at least 5 times higher produced, as we see here by our quantitative mass spectrometry data. And that's just by general biomass. So if you would like calculate this to like cell numbers, it's probably orders of magnitude higher produced. So this really like brought up the question, what like influences Xanthomonas in its natural environment to like make this at such higher concentration, and vice versa. Also, why is Xanthomonas like making this very special metabolite actually like in sugarcane plants. So then after I finished my PhD, I moved on to conduct a postdoc through stay with Peter Dorstein, and like thinking about projects and writing a proposal for the German Research Foundation. I could not come up with anything more exciting than marine microbial communities. And thinking at those dynamics between like the bacteria, the microbiome, and like here, for example, like a diatom, it's really fascinating to me how chemistry and especially specialized metabolites modulate those interactions. And while we have like some synergy on like the left side here, for example, through like the production and uptake of siderophores for iron acquisition, or like metabolic exchange between like different primary metabolites and then vitamins, we have also parasitic activity on the other side. So some of those bugs eventually produce toxins or even like proteases to actually lyse that organism, and then like nourish of the released biomolecules. And what I think is the cool thing for me about this is to think about like the scale. So while this is happening at like the micrometer scale, when we zoom out and think about how those dynamics actually like influence our planet and how small molecules are responsible at the end for planetary interactions, like the global carbon pump. This is really amazing, and I wanted to like I don't know, contribute and get some more insights in these questions. So I think around like the same time was also that I learned about the Tara Ocean Expedition, and from like the first time I heard about it, I thought that's so cool. Like that's probably like one of the most comprehensive metagenome data sets, and it gives us like really like the first time like some insights in like the microbial communities that govern the oceans. What I was wondering at the same time is, like, while I learned more about the expedition and learn more about it, I was wondering still what is the chemistry between those microbial communities. And to me, that's still one of like probably like the big questions that needs to be answered. What is the composition of the ocean's exometabolome? So like all the small molecules. Is this what I just showed you relevant at large scale? And then how does that is connected to the chemical dynamics? So how does that change over time and space? And ultimately, how is that correlated to different ecosystems, and in particular to different microbial communities? So now a lot of research has been done to study small molecules in the ocean. So there's been comprehensive like chemical analysis platforms to resolve dissolved organic matter. That's typically like the totality of all like organic molecules in the ocean, where people sample and then typically acidify those samples, eventually filter them before, and then solid phase extract them with a hydrophobic resin before they then submit them to mass spectrometry analysis. And in case you're not familiar with the mass spec analysis, this is basically just taking all molecules in the sample and separate them by their molecular weight, while plotting the molecular weight against the relative intensity or intensity of the different molecules, and then you get such a mass spectrum, we call it. And as I said, this has been done a lot for dissolved organic matter with ultra high resolution mass spectrometry, so like big machines with huge magnets and incredible high resolution. And I think this is exemplified when you start zooming in at each of those nominal masses here and start looking into detail. You see that there's actually like a multitude of different peaks at each nominal mass, right? So it's really complex. In fact, it's probably one of like the most complex samples we've run in Peter's lab, and now also in mine. And what baffles me is still like the vast like majority of like the unknown component of this dissolved organic matter pool. So what you can do with this ultra high resolution mass spectrometry data, because it's measured so accurately like the different molecules by mass, by the mass defect of like the different elements, you can actually start calculating molecular formulas. And now this has some, of course, uncertainty because eventually different combinations would yield the same mass. But however, if you would pick the right one at the top, of course, there is still like a multitude of different metabolites that can have like the same molecular formula. So while I think direct infusion FT-ICR mass spectrometry, like the gold standard in DOM analysis, is really like a great tool to study elemental flux and look at like fingerprints of these systems. For me as a natural product chemist and an analytical chemist, that's of course like not super awesome because I want to like get like structure to like each of those masses assigned. And yeah, this was one of, I think, the main projects during my PhD to start like using actually tandem mass spectrometry to go like a step farther of like, molecular annotation of DOM. So in case you also don't know about tandem mass spectrometry, so what we do here is we also separate our molecules in a given sample and then isolate different masses over this mass range before we then submit those isolated particular molecules with a certain mass to gas phase fragmentation experiments, where we basically activate them by colliding them against neutral gas molecules, before we then acquire a second mass spectrum of like the fragments now of these molecules to then, again, display this in such a intensity versus MZ mass spectrum. And now the cool thing is that this happens in a very, let's say, typical form of depending on the structure of the molecule. So if you have here a hypothetic molecule, this would like break theoretically with every MS experiments in a similar fashion, and you can somehow like either like predict or interpret like the fragments and build up the structure of these molecules. So now at large scale, this is of course really hard, and I think it's still a super big problem to solve structures completely de novo. But fortunately, our spectral knowledge in like the public domain is growing, and we can actually take this mass spectrum as a fingerprint, and then very similar for like as in sequence alignment, compare this to like a library, and then match the spectra against known reference compounds. So now, while it took us quite some while to like get good quality data from DOM, we I think can now like do this in a regular fashion. But probably like almost every other metabolomics experiment, like one of like the big problems is simply like the amount of spectral knowledge we have, and the vast majority of like the signals we detect remain unknown or unannotated. So, and that's the next technical thing I want to quickly show you, which I think was really a game changer to me when I first learned about it. And this was here, this paper from Jeremy Watrous and also Theo Alexandrov and others in Peter's lab at that time that introduced molecular networking. And the basis is here that you make as well like the assumption that like a given molecule in an MS experiment fragments in a like logical fashion, like here the green and the yellow fragment, for example. And now if you have a very similar molecule that has here modification at one of these carboxylic acids, for example, then the yellow fragment would be shifted by exactly this mass of this modification, as well as the precursor, of course. And now we can make this to our, assumption to take this to our advantage. And simply when we now match experimental spectra against the library, allow offsets between like the different fragments that would correspond to the difference in the precursor mass of those. So, and that's how we can like boost annotation rate probably 5 to 10 fold. And when we typically have 5% annotation rate and now get to 50, this is really a lot. The other thing where I think this is really cool is because we can use the very same algorithm to match the experimental spectra against each other and then organize this information in a molecular network. So here, if we have a set of experimental spectra, depending on their spectral similarity, we could connect them and represent them here as such nodes, and then propagate the annotations between them. So here, if we have an example network, we could say, hey, maybe we don't know like the IDs of all of those molecules, but they all have like very high spectral similarity. So maybe that's a group of lipids or amino acids, peptides, polyketides, and so on. I think for especially natural product research, but also now for I think non-target metabolomics, that's really appealing because I think it's like one way to address the challenge of like unknown chemical space. So now over the years, we refined this pipeline a little bit and plugged it basically in some common metabolomics feature finding tools that integrate the MS1 information and provide you peaks and more accurate relative abundance. So together that really, I think, like builds up the foundation for all our analysis we do with non-targeted metabolomics data. Importantly, besides the analysis tools, so the platform we're using, GMPS, is also a community platform, and it has both like a spectral library as well as like a data repository for experimental data. And that is, I think, the other way, probably in the long run, the even more important aspect of it, to how to address like the unknown molecular space by simply having a user-based, community-based acquisition of chemical and spectral knowledge. So now we also put in some efforts to make this more user-friendly, as we realize that often there's quite some entry barrier to like learn about like all that tools. And I think we're very close to have like a full data analysis solution in the web browser. So we can just take some data from like a spectral, from a data repository and process it on the server in the web browser. And I think that probably makes quite an impact in bringing this analysis to also like people starting with mass spec and metabolomics analysis. However, together, this really is the foundation of like all the data I'm going to show you now in the next couple of slides. And while being at UC San Diego, I think it was not only awesome to be in one of like the pioneering labs in mass spectrometry and metabolomics data analysis, but also working at Scripps Institution of Oceanography, where I had like very close collaboration. In fact, one of my mentors is a professor there, which provided a lot of opportunities to actually get real samples. So over the 5 years I spent in San Diego, there was quite a lot of cruise opportunities and different field trips. Unfortunately, I couldn't go to all of them, but some of them. And we got it like quite a nice amount of actual samples. So we started here at Scripps Oceanography, where we collected like our first samples for method development. Then I went for a couple of weeks to Moorea to like study the chemistry of coral reefs there. We've been mapping stromatolite pools in South Africa, and I also was fortunate to like be on a cruise for a couple of weeks in an offshore environment in the California current ecosystem. That's what I want to focus on for the next couple of minutes. So in case you don't know it, the California current ecosystem is a very productive Eastern Bond upwelling system, where through a process called Ekman transport, where we typically have like strong winds coming from the north, we get like an upwelling system that brings nutrient-rich water from the deep up to the surface ocean close to the coast. And then this nutrient-rich water typically goes like in a current like offshore. And then of course paired with sunlight, like all the biology is happening, and we have like a lot of like primary production and all sorts of sea life going on. And as I said, I was fortunate to spend 5 weeks on the Roger Revelle. This is one of Scripps research vessels where we actually followed such like an upwelling system, which is called Lagrangian sampling. And for me, as a more natural product chemist or now a metabolomics scientist, this was a really nice opportunity because it combined a longitudinal sampling with like a spatial sampling campaign. So here, when you look at the cruise plan, you have to imagine it like we deployed some drifters close to the shore where like the upwelling started, and then followed the system over a couple of weeks here, like in this blue dots, which we call like here a cycle, in which we basically moved with this water body and could like sample every day and get like an idea of how the dynamics are there. And then in addition to that, we also did some spatial transects to basically get like a cross section of what we thought would be like such an upwelling filament. So in total, we visited 26 stations at which we took metabolomic samples. And then, we took different water depth from like the typical CTD cast. So this is such a rosette. They let down at the big steel wire. And then we also get like a depth profile at each of those stations. So then, we took typically 1 liter of water, acidified it, extracted it by our hydrophobic resin, before we then, once back in the lab, submitted it to liquid chromatography tandem mass spectrometry. So here, the important thing I didn't mention before, we first decomplex also the metabolites by a chromatographic step to like deal with like the sheer complexity of it later. Anyway, we got thousands of spectra per run, like at the end, like millions of spectra, and we put them all through our feature-based molecular networking pipeline to then get basically like a big table, which we call like a feature table that has both like relative intensities over all the samples, as well as like connectivity in the network and some different levels of annotations, which include molecular formula, potential like library hits, but also like analog hits. That's when it basically like hit against a library spectra, but with a given mass offset. So now the first question, of course, we were asking is how different like the environments. Can we actually see from like the fingerprints we get like that anything is happening there? And like, yeah, typically like the very first analysis we do is some unsupervised multivariate statistics. In this case, a principal coordinate analysis with Pray-Curtis distance. And if you're not familiar with it, what this basically does, it displays the compositional similarity of multiple samples based on the distance here in this like dimensionally reduced space, either here like on 3 axes or often also 2 axes. And what you have to take in mind is that the closer 2 samples are here in this plot, the more similar are they on their composition. And the more far away, the more dissimilar. And yeah, when we first plotted like the surface ocean samples, so this is now a subset only of like the first 30 meters, where we assume most of like the photosynthetic activity might be going on. It was really nice to see that we see already some type of clustering. So here I color coded the nodes basically on the date when we collected them. And as I mentioned before, we were like starting close to the shore and then following this upwelling system. And yeah, have kind of like a spatio-temporal trend here. And I think this is nicely displayed. So if we would connect all these dots now by the time and the position, we could actually see that in like roughly, we kind of have a trend where we can track this development in this principal coordinate space. At the end here, there were some like irregularities, but I'll get back to that later. However, as like our unsupervised analysis shows like nice separation of those samples and also like in somewhat like explainable fashion. Next, we did some supervised analysis with random forest, where we basically asked the questions, okay, what are like the driving features between those different cycles here? And then, yeah, we kind of like get a ranking of like the different molecules we annotated. So this is now only a subset of the ones which had level 2 IDs. So we're pretty certain about their identity. And it's, yeah, then it's really fun as a natural product chemist to like look at them and look at structures and then eventually find some that are maybe biological meaningful. Like here, homoserine lactones that are like involved in quorum sensing, or yeah, here a manoalide, which is a marine natural product that has been described in sponges, but also like some other toxins that I actually did not thought that they would be there, like diacetoxyskirpenol, which is a fungal toxin often found in food, and so on. Nevertheless, this is just like one way of looking at it. And what I was also wondering is, okay, can we like combine all this information, like only one number to kind of like map that progression on a chemical level? And now here I have to go again a little bit into like technical details. So besides MS matching against our library, we also use in silico approaches where we try to make sense out of like the fragmentation pattern and then get high confident molecular formula. So this is all done through like some software developed by the Berkeley lab in Jena, and I'm a big fan of this tool too, because yeah, I think it gives us like really like high confident molecular formulas from like the MS spectra. So maybe less uncertainty than only by exact mass. However, with like this molecular formula, what we can do, depending if we assume that like certain elements are typically found in as certain functional groups, we can calculate one thing that is called like the average carbon oxidation number based on this formula, which just gives us basically the oxidation level of a feature like for the average carbon in there. And that becomes really cool when we plot this over like all features. And here in this bubble plot, I also included the intensity So basically have the sum product of all of the average carbon oxidation states of my feature table. And then we can build the average. And nicely, we see that there's actually like a decrease in average carbon oxidation here in that first cycle. And when I now map the temperature in the other axes, I think that from a chemical oceanography perspective starts making more sense because, yeah, as I said, there's upwelling. So we start with really cold water. Here was like the black crosses. And then here the red box plots show that at the beginning we have like a drop in average carbon oxidation state. Then we kind of like here have it like plateau. And then as the temperature starts rising again, like it increases again. And what that means, or what, well, my interpretation is of that is that at the beginning, when we have high primary production, like we basically decrease the carbon oxidation state. So like molecules become more energy rich. And then as it moves on, like, yeah, like the primary plume is like over. Now like the heterotrophs are starting to take over and they're like chewing up like all like the labile dissolved organic matter and then like oxidizing it back to a higher carbon oxidation state. So that's one way. And then ultimately what is also really satisfying, I think, is just to look at the molecular networks of this, of this data set. So here, this is now all the information combined from this particular cruise and such a molecular network. And it's important to note that each node, so that's basically a feature or an MSMS spectrum, has also the relative abundance of like the different cycles in it, plotted here as a pie chart. Here, cycle 1, so that's close to the coast, cycle 2, and so on, have basically like these different colors. And now simply by investigating the network, we can see that there's like certain patterns and certain nodes have like a predominant color from like one of those cycles, which like is indicative that they might be like most produced during like a certain spot in our campaign. Here, for example, Manoa-Light, which I mentioned before, which also like ranked highly in our random forest classification, is actually connected to an entire family of different derivatives. So this is really cool. And now we can like go farther and also have like some like chemical or like knowledge-driven exploration where we like look for molecules that we actually know and we think they're like environmentally important. And yeah, we can like explore the chemical space around them. And one I want to particularly highlight is domoic acid, because this is a toxin produced by the diatom Pseudonychia, which has quite some like environmental health implications, both for humans, but also generally for sea life. And yeah, we had a Pseudonychia plume, or perhaps a Pseudonychia plume, and definitely some particular spots in our survey where we found domoic acid at higher levels. And here was a tool actually also from Theodore's lab, Ili. We could like nicely map this on here like our spatial map, and then you would see nicely that actually domoic acid concentrations are like the highest close to the shore, so like cycle 1, and then at these 2 particular spots during one of like the spatial transits. So there it seems that like something that produces domoic acid must have been like pluming at that different stages. Now the cool thing was we know already what makes domoic acid. As I just said, it's Pseudonychia. So this was a really cool kind of like ground truth for us to test the combination actually with like the amplicon sequencing data. Fortunately, some of my coworkers were acquiring at every spot we took the metabolomic samples and tried to bring like the 2 together. So yeah, here looking at the ATS amplicon sequences, we plotted here the like the most abundant OTUs, and in purple this is Pseudonychia. And when you look here in detail, you will see that fits very nicely to the surface waters. Here in cycle 1, that's on the right here, look close to the shore, and then in those particular spots during that transect here. So now that, as I said, was really cool, and we can like correlate this now against like the OTU against like the feature, and then we eventually get some high-ranking correlation score, and could now try and combine this in a new network where we now also display our OTUs as dots, and then add like a new type of edge that's based on like our correlation score. And that becomes very exciting when we do this systematically for like everything, pairwise correlations, and then display this in a new network. And now here, when you look at it from the far, what to me became like immediately apparent was the color coding, right? So like you see here, like a red cluster, then there's green, then there's blue, purple, and then there's also like a mixed one. Now, if we even go farther out and like compare this to our clustering in principal coordinate space, coordinate analysis space, that fits very well to the different clusters here. And now I have to actually solve the thing with like that samples at the end. So like at the very end of our Lagrangian sampling campaign, we actually got into like some eddies where like things were like a little bit perpetuated. So actually does make sense that we kind of go from the left to the right and then back. But nevertheless, I think the important take-home message is here that we can like now start building hypotheses where potentially certain molecules co-occur to certain OTUs, and we can start like now building up on that. And here I found it very interesting that this fungal toxin diazoxyskiprenol I mentioned before actually had like a very good correlation to this like fungal OTUs we fortunately also picked up through our 18S amplicon sequencing. And then even now another derivative. So then we can basically now start building first of all the structure like the microbial community. Like here, for example, Prochlorococcus. So this is more in the oligotrophic ocean. This is small cyanobacteria, and then correlated first against other OTUs to like build like what we think the microbial community could be, and then map the chemical space within that microbial community. Now, it's really exciting to see that. And here, one of the molecules that co-occurred is one of the homoserine lactones that might be produced by some of the heterotrophous bacteria within that microbial community. Or also really interesting, manoalite, again, that has been described as a marine natural product, mainly found in sponges in coral reef. And of course, this brought up immediately the question: is this actually made by the sponge, or could there be such like high chemical similarity between like some offshore community, microbial community, and sponges, or might it be produced by actually not the sponge itself, but one of like the cyanobacteria that the sponge has as a symbiote? Of course, we can only make these assumptions or like hypotheses if there's some sort of like chemical knowledge we find in the literature. So fortunately, a lot of work has been done about homoserine lactones and about manoalite, and simply by Googling the compound names, we can build this hypothesis. But I think what the big take-home message for me was during my postdoc is that the vast majority of molecular space still remains unannotated. And even if we get some putative annotations, we have no idea what they're actually doing. So, and this is now normally where I would start showing you like my research we're doing now in Tübingen. So here we're building a functional metabolomics platform where we try and combine non-targeted metabolomics with some different functional analysis. So one of them is like native mass spectrometry, another is microfluidic devices for phenotyping experiments. But what I thought might be more interesting for you is actually if I talk a little bit more about like the just chemical knowledge that we acquired during like all these campaigns, because I think that might have like some implications for the TRACK campaign you're planning. So having access to like all like this data or like samples is really cool, because what one can do is now start like throwing them all in one bin and try create like some like visualization of like the chemical diversity in all these different systems. And when we run the same principal coordinate analysis of now all the samples we took over the years, it becomes really interesting that you see here clustering of like all like the green dots in this one area. So this is offshore. Then here coral reefs already was quite some variance. But what was striking to me was that like the coastal environments have like such high dissimilarity. So now, of course, there's like a lot of caveats with regards to batch effects, but even within the same sample set, we often see large variance of those ecosystems. And one of the things I really wonder is why is that? And one thing we did in San Diego, I want to quickly highlight, I think gives like a very straightforward answer. There's just like so much change. So when we sampled twice the San Diego coastline here in what we called the San Diego Beach Survey, which also was a little bit motivated by figuring out where like the least polluted water is when you want to go surfing, we saw that in this principal coordinate analysis, if we spot if we visualize all samples between two sampling campaigns, one before a rain event and one after a rain event, that like here, the red ones, that's before the rain, cluster very tightly and there's not much happening. And then when you look at the other data set, the blue one, well, like run through the same data analysis pipeline, actually, well, it's already different, but also we see this huge difference between like those samples collected at the same date. So here, obviously, yeah, there might be like some spatial differences like in the different spots. And when we map this again with the ILI tool, we see nicely that some of the high ranking features that drive here the separation between like the data sets correspond to isoxaben. So this is actually like a pesticide typically used in like gardening in San Diego. And yeah, we can see it nicely here on only the lower map indicated through like the hot colors here, like next to like the San Diego River, but not when it's not raining. And that is very straightforward because there's simply like not much outflow of the San Diego River. And yeah, so we can attribute this to the potential like point source of the San Diego River. Then there were other molecules that did not contribute to separation between the two, like here Igarol. So it only like contributes to like some sort of like spatial variance. Interestingly, yeah, we see like a small effect here after the rain that it like kind of comes out and gets like a little bit washed out here of like the San Diego Marina. And now putting context on that, of course, that is because it's like used as a anti-fouling, a biofouling compound in boat paint. So yeah, basically kills like all like the algae that grow on like the boat hulls. Interestingly, propagating through the molecular network, we could see a very similar spatial pattern for this dicyclopropyl Igarol. That is actually a degradation product that has also been described in some studies that investigated biodegradation through fungi of different like pesticides. However, what I found like really striking, especially while arguing with the reviewers, like we found very little to none actually human-derived molecules. So apparently in the stretches we are like human-derived in a sense of human medications or like typically stuff you would find in a human subject. So rather agricultural pesticides or here like boat paint. And I think that was because there is simply like no point source that has like massive like sewer pollution, for example. And that changed very dramatically when we switched to look like rather at the southern part of San Diego, where we have the Tijuana River, like coming actually like to its delta, and like just flowing out to the ocean. And one important thing is, so it comes basically from the Mexican side, and yeah, it's one of like the biggest I would say like environmental pollution problems in Southern California, because yeah, as soon as it rains, typically like the water treatment plant goes over its capacities, and yeah, like gallons and gallons of raw sewer flowing out into the ocean here. And yeah, one thing we tested was, first of all, what are like the dynamics of this pollution events here in southern, very south in San Diego. And here on this map on the top, you can actually see here the border wall, and then there's like Tijuana on the right side. And we had like these different sample spots here, including one at the SIO Pier, which is farther up north as our kind of negative control, where we did longitudinal sampling during winter when we assumed that there will be some storm events. And now why I'm showing you this is because I think it's really interesting, because in addition to just water samples, we also deployed air samplers, where we were collecting for typically 23 hours just like air through like a GFF filter with like a high flow rate to start looking at the sea spray aerosols that may here exist in the surf zone, where breaking waves create a lot of small bubbles. And when those bubbles burst, they eject tiny droplets, and if the wind conditions are right, they may bring that pollution back inland. So I think people typically just think about, oh yeah, pollution, it's problems, the ocean, don't go swim. But yeah, maybe down there, like in the neighborhoods of San Diego as well as Tijuana, there's also like a potential risk for like people living close to the ocean by just breathing. And yeah, this was the very first pilot study in which we had multiple deployments. Here, one at this Imperial Beach lifeguard tower here with the green dots, and then also one more closer to the river mouth with this red dots. And then, yeah, like the purple dots at the end when we moved the sampling device to the Scripps Pier. And yeah, we sampled for 23 hours, and the protocol was then, besides the air sampling through like this filter devices, very similar to our water analysis, where we did the solid phase extraction for work on, and then we put it in the same tandem mass spectrometry pipeline. And yeah, this was really interesting when we looked at I'm wrapping up, yeah. When we looked now at the chemical drivers and now compounds, the reviewers were asking us before why we did not see them in North and San Diego. They all appeared. Here, different antibiotics and degradation products, other medicines, and illegal drugs. So now, very interestingly, some of them we found mainly here in like the Klubas in the water, but some of them also had incredible high relative intensities in actually the aerosol sample. So that was like a really like mind-blowing moment to me. But why I show you this is because when we now start looking at the temporal trend, and here comparing the water to the aerosols, we see way higher dynamics in the air. And this becomes especially important when we start mapping the wind direction to this. So while here from the left is basically western wind, so offshore, so onshore wind, so wind comes from the sea, and when it comes here from the right, then it comes inland. And like we never had like really like, well, maybe only like 1 or 2 days or so, like clear conditions where we could see like only like collect material from one direction, and then like most of it was mixed or even like only like offshore wind. And now looking at those particular days here for cocaine relative abundance, we saw that there is at least multiple point sources, like both from potential sea spray aerosols, but also massively from city dust. So yeah, then going into the literature, it was also then not too exciting anymore because it has been described in like different like larger cities that you can actually find illicit drugs in city aerosols. So that was something new we learned. But for me, it really like showed like the importance of like all like the sample settings and like the right conditions at the right time, especially for aerosol analysis. So now, yeah, to wrap up, what do we learn from all of this? So yeah, first, I think it's really a nice first molecular inventory of like marine environments, which yeah, here distributed in like this principal coordinate space, or also through like a global molecular network, where we could now go to like all of those different features we detect. Then yeah, we see distinct chemotypes in different marine ecosystems, and often yeah, we see the highest dynamic in coastal ecosystems. And for where we have data, so unfortunately, not all of the studies included like a microbial, like a microbiome component. We can also structure them based on the microbial communities. And last but not least, I think there was not a single environment where we did not detect like some anthropogenic compounds. So I think if we look at like dissolved organic matter and yeah, like metabolome, like yeah, anthropogenics are just like a fundamental part in most of the environments we sample. So yes. Now, of course, like one question is how does that influence the microbial community? So especially when we look at this as a chemical map, often like different compounds are actually maybe chemically similar. We just like classify them as different entities. And yeah, how do those structure microbial communities? And I think, yeah, the TREC expedition could be like a tremendous opportunity to study such coastal dynamics. And yeah, I hope, well, if not through me, that somebody will collect metabolomics data from all those samples, and yeah, start contributing to this global map of chemistry. So yeah, with this, there was a lot of people involved. So yeah, like especially my coworkers in San Diego and everybody in Peter's lab, but also in Lihini Olivares' lab and Kim Prater's lab and Rob Knight's lab, with whom I collaborated over the years, and yeah, like basically helped sampling or provided some additional data products. And now also here my own lab in Tübingen, with whom I gonna start now my more independent scientific endeavors. And then last but not least, like my colleagues in the CMFI cluster of excellence, who have been super supportive so far, and also for like bringing me back to Germany. Yeah, thanks for your attention, and yeah, for having me here. It's a really nice opportunity.
Yeah, thank you very much, Daniel. Let's give Daniel a round of applause, real and virtual, for this wonderful talk, showing a lot of different opportunities how molecules can be captured in a variety of different environments. And actually, I didn't know about these aerosol studies. I think this is really cool. It kind of also provides the link, and also definitely the coastal studies are of huge relevance and interest here. So we'll go now through a round of questions. If anyone has a question, just unmute yourself and speak up. Yeah, Pierre, please go ahead.
Yeah, thanks. Great talk. Just a question I probably missed. I mean, of the tons of peaks you have, what's a fraction where you can put a chemical on it? And that is part one. And part two of the question is, how do you relate it to biology? So you tried to overlay it, for example, with 16S, 18S counts. That's a good starter. But still, it's still association that can be very indirect. For example, if you have a chemical and that group of microorganisms can only be there, not produced by the microorganism, but might be coupled to protozoa or whatever they like and are associated with it, which is not on your radar. So how do you get to biology? That's my question. But first of all, so how many chemicals versus how many peaks? What's the fraction there?
Yes. Yeah, thanks a lot. Yeah, I think I showed it on one of the slides at the very beginning. So we typically get like between 1% and 5% of level 2 IDs. So that means that we get a MS-MS spectrum that matches the library spectrum. So that's kind of like, well, besides authentic standards, the typically highest level of confidence we can obtain. But then there's other levels. So we, like when we expand the library search to like similar spectra that have different precursor masses, but highly matching fingerprints, then we can like boost this to, I don't know, maybe 30, 40%. And then on a molecular formula level, where we calculate now in silico molecular formulas through the fragmentation pattern, it's probably like 60%. Also, but yeah, those are also like decreasing obviously with confidence. And then yeah, for your second question, that's absolutely like on point. Obviously we correlate, especially with like amplicon data, and yeah, like obviously like correlation is not causation. So it really just like provides you like a first structuring of the community. Now, if we have biological knowledge to like follow that up, right? So for example, we know, okay, one is a fungal OTU, one is like a fungal toxin, or one is a toxin that has been described to be made by this particular organism, then that's of course a way stronger hypothesis then. And I think in the long run, like the hope is of course, if we have metagenomes and we can like potentially like get good enough max to like get like gene clusters that we can actually like connect the genes, especially for secondary metabolites that can be like very like specific to the actually secondary metabolite. But yeah, I think there's still like a lot of work to be done to be unambiguous about that.
Yeah, thanks.
Fair. Any more questions from the audience? Oh yeah, Maria, would you like to go?
Yeah, hi Daniel, thanks for the nice talk. Just to follow up on pairs, maybe. So now when you said, right, with this large scale data sets, you can generate some hypothesis on how chemicals and microbiomes in environment interact. Do you plan and how possible it is to follow some of those interactions in the lab? And are you planning to do it yourself or collaborate with someone who has established systems? And what would those systems be?
Yes, totally. I think, yeah, this is absolutely needed. Maybe I should have mentioned that as one of the conclusions So I think, yeah, this is just the first experiment. And then the next thing is to ideally partner with somebody who has like the strains and then follow up in the lab. So for example, like this fungal OTU, we were trying like to find like a strain in the collection at SIO to like prove actually that this makes this compound we saw it, but it was not like a very close match. So I don't know how much like difference there is like in the genus, and it unfortunately did not produce the compound. It doesn't disprove the hypothesis, but of course it would have been nice. So yeah, I think having both kind of like this top-down view from environmental data, but then also having like lab controlled systems, that I think is yeah, what I would do. And unfortunately, as a postdoc, my resources were just like a little limited there. But yeah, I hope that we do more of that in the future. And yeah, partnering with people who understand the systems and who control them very well, who have the strains available. I think this is absolute key.
Sorry. Any more questions from the audience on the valid planetary scale of Daniel's research? Justin, maybe, just to steer it up. Or anyone else? If you feel like you want to just ask questions in the box instead of speaking up, please type them in the box. In the meantime, maybe I'll ask a question. Yeah, once again, super nice talk, Daniel. So in terms of logistical challenges, so if, for example, we're talking about sampling in particular coastal regions, so what do you think will it be like piece of cake, or what will be a particular challenge? Is it like temperature control? Is it like shipment? Is it degradation? Can you go through this?
Mm-hmm. Yeah, I think like infrastructure is key, right? Like, so I was very fortunate to work at Scripps Oceanography, and when we did the sampling at the coast, I was actually like on the small Zodiac myself, and we could just launch this from the Scripps pier, and it was also, I think, yeah, there are some common funds that just pay the gas. We didn't even have to pay that, so that was of course very easy. Now it's different if we had to trailer the boat somewhere, right? And now it becomes even more like difficult if you go to like a remote like environment. So like the work in Moorea was only possible because there's a long-term ecological research site on like, so that was one of the coral reefs that operates like a station there, which like is insanely expensive. And then like the large ship operations on the Roger Revelle, I think it costs like $50,000 a day. So obviously there's like a huge infrastructure and cost associated to that, to which I was fortunate, that we could hop on those opportunities. So I think starting from scratch is probably really hard, and the best thing to do is to partner with existing infrastructure if there's opportunity. Or I don't know, of course, if you get like a massive amount of resources, you can also start this from scratch, but I think it's hard.
Perfect. Yeah, thank you, Daniel. So maybe chance for the last question because Daniel has some program for the rest of the day, and I probably will not abuse our hospitality by saying that if you want to follow up, please reach out to Daniel directly. His main email and contact information is at University Tübingen. It's actually pretty close here. And as far as I know, Daniel travels from time to time passing Heidelberg. So probably he will not, if you were interested and would like to see him. So Daniel, would you be willing to stop by and catch up with any sort of questions?
Sure, I'll be super happy.
Cool.
Yeah, can I ask one really fast question, Theodore?
Justin, your time.
Yeah, sorry. We should catch up at some point, Daniel, because I know Theodore, we've been chatting a lot. Yeah, because I'm kind of interested in terms of, batch effects, processivity, in terms of, how scalable are these, right? Because as we're approaching a lot of these questions, we want to go big, right, potentially. So yeah, if you could just comment on that briefly, and then we can talk more offline. Yeah.
No, thanks a lot for bringing this up. There's of course like a lot of nuances, and we're struggling with batch effects. So one important thing I should probably have mentioned, I think it was written in the caption. So when we do like the sample to sample comparison within a data set, it's not that big of an issue, because we randomize them, we run them in the same batch, it's fine. But then if we want to like compare samples from 3 years ago to like the ones I'll collect tomorrow, that's of course harder. One thing we use to our advantage there is the MS-MS similarity. So now where before we would cluster the different like features over samples by retention time and mass, right? And that I think is a very delicate part. We now cluster them by their MS-MS similarity. And I think this is inherently like more reproducible than retention time. So when I showed you like that big principal Conan analysis here, so this is clustered on MS-MS level. So here now we only like align basically all the features on MS-MS level, and I think this is for large scale more robust. Also, the feature finding is computationally expensive, and I think the scale's better. So if we do thousands of features, I would probably not try to do the comprehensive MS1 peak building. But rather only look at the MS-MS spectra. Similar as in some proteomics approaches where you then get spectral counts. I mean, it's not as accurate anymore, but I think for scalability's sake, I think it's right now the probably best bet.
Okay, cool. All right,
Cool. Yeah, thank you, Justin, and thank you, Daniel. And so I'm actually blown away by the throughput, but also amount of information that you can put together here and still find patterns that is not completely, just driven by batch effects or by days of sampling and so on. Again, from a spec perspective, this is really, really cool to see that mass spec, I mean, in your hands is a very valuable tool. So yeah, with this, I'll probably be wrapping up just to give you 2 minutes, Daniel. So before your whole program starts and so on. Thanks everyone for tuning in. Thanks for your questions. And again, Daniel is in Tübingen, and so just reach out to him if you have questions, and let's give him one more last round of applause.
Thanks, Daniel.
Thank you. And yeah, thanks a lot for having me.
All right. So then we're wrapping up. So Daniel, this was super cool.